Welcome to this session. So in this session, we are going to learn about transport across cell membrane, and this is a part 2 video, which is active transport mechanisms. So yesterday we have seen the passive transport mechanisms and what are the different types of passive transport mechanisms. Today we will learn about active transport mechanism. So this we have seen already, why the transport across cell membrane should be there. What is the importance of this transport across cell membrane? So the nutrients must be transported into the cell. At the same time, metabolic waste products must be transported out of the cell. So cells achieve these mechanisms by different transport mechanisms, and this also we have seen already. So transport mechanisms are classified into passive transport, active transport, and vesicular transport. Already we have studied detailedly about passive transport mechanisms, and today we will learn about active transport mechanisms. So what is active transport? What is passive transport first? So you know that passive transport is transport of substances along the gradient without expenditure of energy. So what is active transport? So it is opposite to that of the passive transport. So transport of substances against the gradient with the expenditure of energy. So this you have already seen this picture, and here we have both the type of transports. So passive transport, so here you can see passive transport, we know there is two types, diffusion and osmosis, and again diffusion is of two types, simple diffusion and facilitated diffusion. You know, in simple diffusion, in both of these passive transport mechanisms, substances from higher concentration, they'll move towards the lower concentration. So they are moving along the gradient, and there is no energy, energy is not required for the transport of substances here. So this is a passive transport mechanism. And here today we are learning the active transport mechanism. Here you can see from lower concentration of substances, the substances are transported into higher concentration area. So here we need ATP for the transport of substances. So this type of transport is called active transport. So what is active transport? It is nothing but the transport of substances from lower concentration to the higher concentration, that is against the gradient with the expenditure of energy, and it is also called as uphill movement. So this active transport is also carrier mediated. So here you can see, yesterday we have studied about facilitated diffusion. I told you the other name for facilitated diffusion is carrier mediated diffusion, because it uses carrier protein to transport substances. In simple diffusion, because it is the lipid soluble substances, they will simply diffuse through the cell membrane, whereas the lipid insoluble substances, for example glucose and amino acids, they will be transported through the carrier proteins from higher concentration to lower concentration. So with the help of carrier protein, they are transported. So that is why it is called as carrier mediated or facilitated diffusion. Here also you can see, for the transport of substances from lower concentration to higher concentration, we need carrier protein. So again, it is a carrier mediated type of transport, but here we need energy. So it is a type of active transport. So this carrier proteins are called as active pump mechanism in active transport. So the substances which are transported through this active transport are ionic substances like sodium, potassium, calcium, chloride, iodide, even hydrogen. All these are transported by means of active transport, whereas non-ionic substances like glucose, amino acids, and urea can also transported through active transport. So we already studied that with the help of passive transport, with the help of carrier proteins, glucose and amino acids are transported. So here also, again with the help of active transport mechanisms, again it is also a carrier mediated type of transport, but with the expenditure of energy, again these glucose, amino acids, and urea, they are also transported through this mechanism. So this active transport is of two types. Active transport is of two types, which is primary active transport and secondary active transport. So active transport is nothing but it is the transport of substances against the gradient with the expenditure of energy, which is uphill movement. So what is the difference between primary active transport and secondary active transport? We have seen in the picture that to transport substances from lower concentration to higher concentration, we need energy, and in primary active transport, the energy is directly liberated by the breakdown of ATP, which is liberating the high energy phosphate compound. When it breaks down, ADP and phosphate is forming, and this provides a energy. In case of secondary active transport, when the primary active transport utilizes energy and there is some form of energy which is stored in the form of ionic concentration difference in the cell membrane. So transport of one substances will produce this ionic concentration difference in the cell membrane, which is utilized for the transport of some other substances, and this type of transport is called as secondary active transport. We will study detailedly. And the primary active transport, I told you, it is the energy is directly derived by the breakdown of ATP or some other high energy phosphate compounds, and this carrier mediated and active transport mechanisms are called as pump mechanism, active pump mechanism. We have seen that. So here we have the primary active transport mechanisms in our body. So sodium potassium ATPase pump, calcium ATPase pump, and hydrogen potassium ATPase pump. These are the examples for primary active transport which is happening in the body. So sodium potassium ATPase pump, this you all know, it is a very important question in exam point of view. So there are many MCQs can come and even 5 mark can come from this sodium potassium ATPase pump. So sodium potassium ATPase pump, calcium ATPase pump, and hydrogen potassium ATPase pump. So we will study detailedly about these pumps. So this sodium potassium pump, it is present in all the cells of the body. So in all the cells, the cell membrane will contain this pump which is sodium potassium ATPase pump. It will pump, you can see here in the cell membrane it is present, and it will pump 3 sodium outwards and it will pump 2 potassium inwards. So it is involved in the efflux of 3 sodium ions, influx of 2 potassium ions. So the coupling ratio is 3:2 here. So it is an electrogenic pump. Why it is called as electrogenic pump? This is very important to explain. So here you can see 3 positive ions moving outwards and 2 potassium ions moves inside. With the help of this sodium potassium ATPase pump, 3 sodium ions moves outside and 2 potassium moves inside. So what happens when there is high amount of positive ions are pumped out and only 2 positive ions are pumped inside? So what happens inside, there is less positivity, or we can say there is negativity. So this creates an electronegativity inside the membrane, and this is why this sodium potassium is called as electrogenic pump. Whereas 3 high amount of positive ions are, whereas you can see 3 positive ions are pumped out and only 2 positive ions are pumped inside. So what happens inside, there is a less number of positive ions, or we can say there is negativity inside the membrane. This is the one reason which is involved in the generation of membrane potential inside the membrane. So inside the cell membrane is negative at rest. This is due to the presence of sodium potassium pump in the cell membrane. And this is one of the most energy using process in the body, on an account of 24% of energy is utilized by cells in the body, whereas in neurons it can be account for 70%. So it is the most energy using process in the body, this sodium potassium ATPase pump. So coming to the structure of a sodium potassium ATPase pump, so a sodium potassium pump is having 2 subunits, one alpha subunit and one beta subunit. So these two are both are different. So you can see structurally they are different, and that is why this sodium potassium ATPase pump is called as heterodimer. It has one alpha subunit, one beta subunit. This beta subunit is just a glycosylation sites, whereas the alpha subunit has extracellular binding sites and intracellular binding site. There are 3 intracellular binding sites and 2 extracellular binding site. What are the 3 intracellular binding sites? We can see 3 sodium binding site, phosphorylation site, and ATP binding site. In the extracellular, we have 2 potassium binding site and there is ovabin binding site. So these 2 are the extracellular binding sites. So we'll study one more time the structure of ATPase pump. So ATPase pump, sodium potassium ATPase pump, it is present in all the cell membranes. It is having 2 subunits, one alpha and one beta subunits. Both are different, and that is why it is called as heterodimer. And the beta subunit is attached with the glycosylation sites, whereas alpha subunit has 2 extracellular binding sites, which is 2 potassium binding site and ovabin binding site, and 3 intracellular binding site, which is 3 sodium binding site and ATP binding site and ATP phosphorylation site. So this is the structure of sodium potassium ATPase pump, and how it will occur. What is the mechanism of this sodium potassium ATPase pump? So what will happen when 2 potassium and 3 sodium binds with this extracellular and intracellular binding site, what will happen? This sodium potassium pump, the ATP will be phosphorylated into ADP and phosphate. This phosphate, which is the high energy compound, which will make the sodium potassium pump to change it, to change the shape. That is, the conformational change will occur in the sodium potassium pump, and that is why 3 sodium will be pumped out and 2 potassium will be pumped in. So this is how the sodium potassium pump works. So what is the function of sodium potassium pump? It mainly controls the cell volume. So it is the most important function of the sodium potassium pump. Otherwise, without this sodium potassium pumps, what will happen to the cells? Cells will swell and they'll burst. So to prevent this, why cells will swell and burst? Because the sodium, if there is no sodium potassium pump, what will happen? The inside of the cell membrane, we have studied in the ionic composition difference in the extracellular and intracellular fluid when we studied about the body fluid compartments, the ionic composition, right? So what happens? Inside of the cell membrane there is more of proteins, and outside the cell membrane there is more sodium chloride bicarbonate ions. So because inside there is more proteins are the anions, which are negatively particles. What will happen? If there is more negative ions, they'll try to attract, or they'll try to absorb the positive ions from the high concentration side, that is sodium, potassium, and the other positive ions they'll try to attract, and with the sodium and other positive ions, the water will also move, which can cause osmosis of water. So what will happen? If the positive ions moves inside, because of the osmosis of water, the cells will swell and they'll burst, and this is prevented mainly by this action of sodium potassium pump. So sodium potassium pump controls this cell volume, and this prevents the osmosis of water into the cell, and this prevents the swelling of cell and the bursting of the cells. And the second function we already know, it is an electrogenic pump. So because 3 sodiums are pumped out and 2 potassium is pumped in, the electrogenicity is created, and this is the reason why there is electrical potential across the cell membrane. So this is very important for the function of to transmit the signals in nerves and muscles. So this sodium potassium pump, the activity of sodium potassium pump can be increased or decreased by some factors. So the factors which increases the sodium potassium pump activity is like cyclic AMP, which is cyclic adenosine monophosphate, and diazyl glycerol, thyroid hormones, aldosterone, insulin, and G-actin. One thing we can remember is the cell membrane of thyroid hormones will have more number of sodium potassium pump, and the thyroid hormones will increase the thyroid hormone will increase the number and activity of the sodium potassium ATPase pump in the cells, and because of this, normally thyroid hormones will increase the basal metabolic rate. One of the function of thyroid hormones is it will increase basal metabolic rate, and it is also due to the increasing the activity of sodium potassium pump by the thyroid hormones. So factors which will decrease the sodium potassium activity is low temperature, hypoxia, substances like dopamine and oabin and related substances, and the drug like digitalis. And this digitalis is used in the treatment of heart failure, because it will increase the myocardial contractility. So that is why digitalis is used as a drug in the treatment of heart failure, whereas this digitalis will decrease the activity of sodium potassium ATPase pump. This is one of the clinical aspects we should remember. So this is all about sodium potassium ATPase pump. We will revise the sodium potassium ATPase pump in the summary. So I'll come to the next pump, that is calcium ATPase pump. This is also a type of primary active transport mechanism. So here you can see this is the structure which you will study when you study excitation contraction coupling or the muscle contraction, skeletal muscle contraction. So here you can see this is the structure which is showing the sarcotubular system. You know that in the sarcotubular system we have, this is the cell membrane which is sarcolemma, that is the muscle membrane. The invagination of sarcolemma forms the T tubules, whereas the longitudinal sarcoplasmic reticulum forms the L tubule, and this, you know, this is the triad which is having 2 terminal systems and 1 T tubule, right? So this forms the triad. And here you can see what is the function of this sarcoplasmic reticulum in the muscle. It will store large amount of calcium ions, and when calcium ions are released from this sarcoplasmic reticulum, this will involve in sequence of events which will involved in the process of muscle contraction. Once the muscle contraction is over, what will happen? These calcium ions will be pumped back into the sarcoplasmic reticulum through the pump which is present in the membrane of the sarcoplasmic reticulum, and the name of the pump is called Serka pump. What is the name for this pump? Serka pump, which is explanatory: sarcoplasmic endoplasmic reticulum calcium ATPase pump. So what is this? It is present in the membrane of the sarcoplasmic endoplasmic reticulum, so you can remember easily. That is sarcoplasmic endoplasmic reticulum calcium ATPase pump, and this pumps the calcium back into the sarcoplasmic reticulum, which is very important for the process of muscle relaxation. So this is one of the example for calcium ATPase pump. Actually, the calcium pump is also present in most of the cells in our body, which is also very important for maintaining the low intracellular fluid calcium concentration. So this is one of the example which is present in the skeletal muscle. And the next pump mechanism is hydrogen potassium ATPase pump. This is especially seen in the parietal cell. The very good example for this hydrogen potassium ATPase pump is it is present both in parietal cell as well as in the renal tubules. It is this hydrogen potassium ATPase pump. It will present in the parietal cells as well as in the renal tubules. Here I have shown the example which is showing the parietal cells. And in the parietal cell, this is the parietal cell which is having the luminal side. This is the apical side, and this is the basolateral side. In most of the cells, basolateral membrane contains this sodium potassium ATPase pump. You know, this is also a primary active transport mechanism which will pump 2 potassium inside and 3 sodium outwards, right? So this is the function of sodium potassium ATPase pump. This is present in the basolateral side of the parietal cell, and here you can see the apical side of the parietal cell will have this hydrogen potassium ATPase pump. For the other name for this hydrogen potassium ATPase pump is proton pump in the parietal cell. Proton pump. So the other name for hydrogen potassium ATPase pump, and this is also a primary active transport mechanism which will pump hydrogen into the lumen and potassium into the cell. So this helps in the the secretion of hydrochloric acid. This pump helps in the secretion of hydrochloric acid, where the chloride ions will be transported into the lumen along with the hydrogen ions, and there are many mechanisms which is involved. So hydrochloric acid will be formed in the lumen, which is a very important function of stomach, you know that. So parietal cell will help in the formation of hydrochloric acid, and this pump help in the secretion of hydrogen ions into the lumen. So hydrogen potassium ATPase pump, this is also a primary active transport mechanism. This is the mechanism which is seen in the parietal cell. And this you will study detailedly when you study the GAT, when you study the stomach, that is especially the gastric juice secretion. This pump can be inhibited by drugs. So hydrogen proton pump inhibitors, and which is used in the treatment of peptic ulcer. So secondary active transport. So that's all about the primary active transport mechanism. Coming to the secondary active transport, I told you secondary active transport is, here the energy is derived secondarily from the energy which has been stored in the form of ionic concentration differences between the two sides of the membrane, which is created in the first place by the primary active transport. So here the substance will be coupled with the active transport of sodium. So that is why the secondary active transport is classified into two types, which is sodium co-transport and sodium counter-transport. I told you the transport of substance will be linked with the transport of sodium. That is why secondary active transport is of two types, that is sodium co-transport and sodium counter-transport. We studied this type of transport in the facility diffusion, where we can see we have seen there are three types of carrier proteins which are called as uniport, symport, antiport. I told you symport and antiport are together called as co-transport mechanism. So here we have sodium co-transport and sodium counter-transport. So here you can see the sodium co-transport mechanism. Here we have a carrier protein which is similar like and symport, where we can see two binding sites in the carrier protein. here you can see the example sodium glucose co-transport, where one binding site for sodium and there is another binding site for glucose. So when the sodium is transported, along with that glucose is also transported, and this is also seen in the intestinal cells as well as in the renal tubular epithelial cells, which is helpful in the reabsorption of glucose in the intestine as well as in the renal tubular epithelial cells. And the sodium counter transport, this is here the carrier protein acts as antipode, where when the sodium is pumped outside, when the sodium is pumped inside, the calcium is pumped outside. So this is the pump which is called as sodium calcium antipode. There is one more example where when the sodium is pumped inside, hydrogen will be exchanged outside. So this is sodium hydrogen counter transport. So this is sodium calcium counter transport. This is sodium hydrogen counter transport. So this sodium calcium counter transport is present in most of the cells, whereas this sodium hydrogen counter transport is also seen in renal tubules. So coming to the MCQ session, which of the following is an example of secondary active transport? So secondary active transport is of two types, secondary active transport of two types, sodium co-transport and sodium counter transport. So all these are pump mechanism, right? Hydrogen, potassium ATPase pump, calcium ATPase pump, sodium, potassium ATPase pump, all these are primary active transport mechanisms. Only sodium glucose co-transport, so SGLT, and we can also say glucose dependent, the sodium dependent amino acid transport. So that is also an example for secondary active transport. So the answer for this question is sodium glucose co-transport. So this is the answer for this question. All the other answers are primary active transport mechanisms. So what is the function of sodium potassium ATPase pump in the cell membrane? So influx of 3 sodium and efflux of 2 potassium, efflux of 2 sodium, efflux of 3 potassium, influx of 2 sodium, efflux of 3 potassium, efflux of 3 potassium, efflux of 3 sodium and influx of 2 potassium. Which is the answer? Yes, the answer is D, which is efflux of 3 sodium and influx of 2 potassium. So the answer is this one. So 3 sodium will be pumped into the extracellular fluid and 2 potassium will be pumped into the intracellular fluid. So sodium potassium ATPase pump moves 2 potassium ions to extracellular fluid and 3 sodium ions to intracellular fluid. So this is wrong. This is not true. It is important for controlling cell volume. Yes, it is true. Sodium potassium pump controls cell volume. Otherwise, cell will swell and burst. It is an ion channel, all of the above. No, it is not an ion channel. Here we have the carrier proteins, and it is not true. So the answer for this question is it is important for controlling cell volume. So we can summarize the functions of sodium potassium pump as it is an electrogenic pump. It is the one which is controlling cell volume. So these two are the very important functions of sodium potassium pump. Sodium potassium pump can be inhibited by all except if concentration of sodium potassium ATP is too low, when temperature is reduced, if plasma concentration decreases, oxygen lag. So we have seen what are the factors which is decreasing the which will inhibit the sodium potassium pump activity. That is low temperature, hypoxia, drugs like digitalis and ovabin related substances, dopamine. All the substances will inhibit the activity of sodium potassium pump. So the answer is here, if the plasma calcium concentration decreases. If the plasma calcium concentration decreases, nothing will happen to sodium potassium pump. You know that normally within the cell, intracellular fluid will have low calcium concentration. So this will not affect the sodium potassium ATPase pump. So to summarize, active transport is transport of substances against the gradient with the expenditure of energy, and this active transport is also called as uphill movement, and this active transport is of two types, primary active transport and secondary active transport. In primary active transport, energy is directly derived from the ATP, and in secondary active transport, the energy is derived secondarily from the stored energy, which is in the form of ionic concentration difference in the cell membrane. And the primary active transport, the examples are sodium potassium ATPase pump, calcium ATPase pump, and then hydrogen potassium ATPase pump. And the sodium potassium ATPase pump, it is present in all the cells. The functions are controlling cell volume, and it is an electrogenic pump which pumps 3 sodium outwards and 2 potassium inwards. And the ratio for this sodium potassium pump, the coupling ratio is 3:2. And the structure, if we take the structure of sodium potassium pump, it is a heterodimer structure where we have one alpha and one beta subunit. The beta subunit is the glycosylation site, and the alpha subunit will have two extracellular binding sites and 3 intracellular binding sites. So intracellular binding sites are 3 sodium binding site and ATP phosphorylation site and ATP binding site, and then extracellular binding sites are 2 potassium binding site and OABIN binding site. So what will happen? What is the mechanism of action? When 2 potassium and 3 sodium binds at their binding sites, the conformational change will occur in the sodium potassium pump after the phosphorylation of ATP, and this is how the 3 potassium are pumped out and 3 sodiums are pumped out and 2 potassiums are pumped inside into the cell membrane. So this is the sodium potassium ATPase pump. Whereas the calcium ATPase pump, this is especially the Cerca pump, which is present in the skeletal, the sarcoplasmic endoplasmic reticulum membrane, and this helps in the pumping of calcium back into the sarcoplasmic endoplasmic reticulum, which is very important for the muscle relaxation. The third pump, which is hydrogen potassium ATPase pump, that is present in the parietal cell as well as in the renal tubules, which is helping in the secretion of hydrogen into the lumen. So the secondary active transport is linked with the transport of sodium. So sodium co-transport and sodium counter-transport. Sodium co-transport, the examples are sodium glucose co-transport, sodium amino acid transport. All these are co-transport mechanisms, which is similar like symport, and sodium counter-transport, which is similar like antiport. Here the sodium calcium counter transport and sodium hydrogen counter transport. So these are present in the renal tubules. So that's all for today's class. Thank you.